need to kind of explain some techniques. So when we are talking about COVID-19, and everyone knows that we have been facing this pandemic from past 6-7 months, and it's completely engulfed whole of the population of the world, and because the most important thing about addressing a disease is that how we are diagnosing the disease. Looking for vaccines, and before we start looking for treatments, and before we start looking for managing the disease, the first important thing that we need to address in well by the SARS coronavirus. Again, there was a similar kind of disease caused by a different variant of this virus that we call as MERS, Middle East Respiratory Syndrome. So it was very well expected that scientists were from all over the world who are experts studying these coronaviruses. They had already expected that there could be likely something like a kind of pandemic, a kind of SARS coronavirus variant that might cause some kind of outbreak in the near future. So that was already, expected. Scientists all over the world had already expected that. So when we are talking about a disease outbreak, and this thing has already happened. So back in China, back in December in China, when these things started happening, the scientists started diagnostic procedures and trying to know what this disease is exactly all about. So they had the cases of bilateral pneumonia and similar kind of things that happened back in with SARS coronavirus 1 in 2002. And so here the most important thing was that how quickly were the scientists able to detect this pathogen and develop a reliable tool with a proper specificity and a proper sensitivity to diagnose this particular pathogen. So we all know that this is a virus. It's an RNA virus. So in viruses, we have different kind of viruses. We have a single-strand RNA virus. We have double-stranded RNA virus. We have double-strand DNA virus, and we have single-strand DNA viruses. So based on the nucleic acid these viruses harbor, so they are classified into various categories. So this is a coronavirus, and it comes under the order Nidovirales, and this virus is a simple single-stranded positive sense mRNA virus. And because this is an mRNA virus, the first thing that comes in our mind is whether we'll be able to use the molecular diagnostic tools, those kind of tools that has that are in picture from last 2 decades. Because of these techniques, the advent of various molecular biology tools, we have been able to develop certain techniques like a simple PCR, polymerase chain reaction, a simple qPCR, a real-time polymerase chain reaction, microarray assays, a lot of other assays which we can use to develop certain tools to diagnose any pathogens. So first of all, a very important thing we need to understand is that when we are going for a diagnosis of a pathogen, so we have certain choices with us. So what is the choice with us is the pathogen that's infecting a host. Would we be able to diagnose this pathogen by maybe diagnosing the nucleic acid of the pathogen? So nucleic acid is a very valuable tool, very valuable material of the pathogen, which we can anytime extract and maybe use that nucleic acid for diagnosis of this particular pathogen. So any diagnostic, any infectious disease, the gold standard of that diagnosis of an infectious disease is based on either detecting its nucleic acid or detecting the proteins, the antibodies that are raised against, that are elicited against this particular pathogen, the proteins of this particular pathogen in a host. So these are two important things. One is the nucleic acid. Either we can detect the disease by detecting the nucleic acid of the pathogen, or else we can diagnose the disease by detecting various antibodies that have been elicited against this pathogen in the host. So these are two important factors. So in any case, in both the cases, we need to take the samples. We need to take the samples. So as we know, this is a respiratory disease. So we would expect that this particular pathogen, it replicates in the respiratory tract. So unlike its other viruses, the similar viruses, SARS coronavirus 1 and SARS coronavirus 2, so this virus has been primarily seen to replicate in upper nasal respiratory tract. So the replication is also possible. It has been seen in lower respiratory tract. That means down the trachea, inside the lungs. There have been cases where we have been able to see those things, but the replication has been primarily seen in the upper nasal respiratory tract. So when we talk about the MERS or we talk about the SARS, we would see a good amount of replication in the lower respiratory tract, and we would also see the presence of the viral particles in the blood. So that we call as viremia. So this is something why I'm talking about all these things, because these things are very important when we are talking about the diagnosis of the virus. So we need to understand, if we are trying to diagnose a pathogen, if we are trying to look for a pathogen inside the host, what is the tissue we need to look at? What is the site we need to take the sample from? So that's important. In many viral diseases, we look for the pathogen, we look for the virus inside blood. We can simply take the virus, the blood sample, and isolate the serum from the blood sample, and the viral diseases that cause viremia, that means the virus inside the bloodstream. So we'll be easily able to take the blood and try to diagnose the virus inside the blood, inside the serum. So, but in this case, that's not possible. So the problem is that it does not cause viremia. So you will not see, in very few cases, out of maybe 1 out of 10 or 1 out of 20 cases, you'll be able to see viremia in the patients. And that also when the severity of the disease is very high. So in most of the cases, when you are talking about diagnosed pathogen, we need to look for the site where we take, where we are taking the samples. And since the replication is primarily, it happens in the upper nasal respiratory tract, so the most easiest way to take the sample is to take sample from the nasopharynx. So nasopharynx is somewhere, I mean, somewhere at our throat. So there are two kind of pharynxes. One is the oropharynx and one is the nasopharynx. So nasopharynx is a little bit easier way to take the sample, because we expect that the virus would have replicated and we will have good amount of virus particles that can be seen in the secretions around nasopharynx or around oropharynx. So that's why whenever you might have been seeing the pictures while of sample collection in this SARS coronavirus 2, you will see that people are being, screened for this by through, the sample collection is done through swabs, and you will see those long swabs. They are being put inside the nostrils until it touches the nasopharynx, and we have a kind of three screwing movements at that place, so that the amount of secretions, they get kind of stuck to the swab, the cotton swab, and we are able to take the swab and put in a medium, the virus transport medium that we call as. So it can also, we can also use the oral pharynx, and for that we need to use the oral pathway and make sure that the swab touches the oral pharynx. But it has been seen that taking samples using the nasopharynx and putting those swabs in the nostrils is a little bit easier than using the oral pharynx. So that's why, I mean, people have been more comfortable in taking the swabs from the nasopharynx. And these collection of these samples have been pretty much easier and pretty much more reliable. And the virus particles, I mean, the virus titers have been found to be better when collected through the oral nasopharynx in comparison to the oropharynx. But in any case, if it's not possible to take the sample from the nasopharynx, the oropharynx is always the possibility. So once we are taking these samples and we make sure that the virus particles are there. So because we need to understand once the virus infects, so it infects through, there's a droplet transmission. And now lately there have been various reports that there is an air transmission. The virus is airborne also. And recently WHO has cleared its, through its statement that maybe there is a possibility of airborne transmission in enclosed spaces. So in any case, I mean, when the virus infection happens, it happens through droplets. It happens through the air transmission. That means whenever a person inhales those virus-laden droplets or the airborne droplets, those probably will go inside the nostrils, and they will probably find the receptors that are lining the nostrils, the mucous membrane, and that's where exactly the virus particles can gain entry inside the cells. So once they gain entry inside the cells, they will start the replication, and with every replication, the viruses will come out and they will stuck to their excretions, lining the nasal mucosa of the upper nasal tract and the nasopharynx, and very well the oropharynx. So you need to understand that. So the virus kind of is very much present in the nasopharynx, in the oral pharynx, and these are kind of more susceptible parts. And so that's why we have been seeing, the use of masks and other things, other protections like face shields. They pretty much play a very important role in breaking this transmission. So that needs to be understood before we think about diagnosing this disease. So, so once we have these samples with us, we have collected the samples. So we would expect that once we have collected these samples, so we have a lot of virus particles that are actually stuck to these swabs, and we are taking these swabs and putting them in a virus transport media. So virus transport media is nothing but simply a kind of buffer that keeps that that protects the integrity of the virus. So it does not kind of destroy the virus. So to make sure that we might have to transport this virus for detection over a longer period of time or longer distances. So that's why we make sure that this swab is put in a virus transport medium so that it maintains its integrity. So, like we said, that we, as we understand this disease is kind of is it shows its symptoms lately, and I think that all of you must have already understood the disease. That once this disease takes place, once the infection takes place, after it shows its symptoms after like 5 to 7 days. So even people have noticed that these symptoms are shown up to 14 days. Some have reported even up to 28 days. They were not able to. I mean, the symptoms were delayed up to 28 days after the infection. So that's why this diagnosis thing becomes a little bit tricky. I mean, once we are diagnosing the virus, I mean, we make sure that only happens when there has been a contact. I mean, if there is a person who has had a contact with a person who already is infected with this virus, that means he will not be have any kind of symptoms up to like 5 to 7 days. So after the contact with an infected person, the person will show its symptoms after like a week or 5 days of the contact. So that's why it becomes a little bit tricky. So in these 5 days before the person shows the symptoms, the virus does replicate. So the titers are a little bit higher. And because that is the case, and in these 5 days we have not been able to detect this disease, or where this because the symptoms are not there, based on the symptoms. So there is every chance that in these 5 days, if this person is the infected person is comes in contact with the other people, so he might very well transmit the disease to the other people. So that's why this diagnosis stuff kind of this becomes a little bit tricky. So we have to instead of kind of relying alone on the symptoms, we have to rely on the contacts. We have to make sure that we make sure that we rely on the contacts, and before the person starts showing the symptoms, the contact person starts showing the symptoms, we go for the diagnosis of the disease. So whether these people are infected with this disease. So that's why it's a little bit trickier, because unlike other diseases, viral diseases, wherein usually the incubation period is not too longer. I mean, the diseases, the viral transmission sometimes don't take much time, and because in this disease, a lot of, I mean, infected people remain asymptomatic. So that means once the incubation period is also longer, and the second thing is that some of the people remain asymptomatic. So that means will not even know that whether the people I mean, that have come in contact or have not come in contact, whether they do have, I mean, the virus infections or not. So that's why, I mean, we have not been able to control the pandemic to the degree that we can do with other viral diseases, because here the thing is that the asymptomatic patients who have been infected but are not showing the symptoms can very well transmit the disease like their symptomatic partners, or the partners the other infected people who have very well the severe symptoms of the disease, where the severity is very high. So that does not affect the transmission. So that's why the diagnosis is a little bit trickier in this part. So in any case, I mean, when we are diagnosing, when we are going for diagnosis of the disease, so we do it from the taking the nasal swabs of the nasopharynx and oropharynx, and once we put that in a transviral transport medium, which I said it maintains the integrity of the disease. So we after that go for a test that we call as real-time PCR. So we need to understand, like I said, that there are two important diagnosis tests. So that is diagnosing a nucleic acid of the pathogen, and second is that diagnosing the antibodies of the pathogen. But because the antibodies, they are kind of elicited after like 7 or 9 days after the infection, or 9 to 14 days sometimes after the infection. So the problem is that we'll not be able to disease if we are going for antibody tests or serological tests. So we will not be able to diagnose the disease for first 9 days of the infection. So that's something, the disease has already occurred, the symptoms have already occurred. Then only will the neutralize. Then only will the other antibodies that we can kind of diagnose, that we can use for diagnosis. Then only those antibodies will start coming. I mean, will start coming in the patients. So we'll be able to see those antibodies in the serum, detect those antibodies in the serum using various serological assays like ELISA and the other antibody assays. But the problem here is that we'll not be able to do that at least before before 9 days. So we have to start it after a week, 9 days, or after 2 weeks. So then only we'll be able to see those antibodies. So that's again a problem. I mean, when you are talking about an outbreak, when we are talking about a pandemic, so we cannot wait for like 9 days or 2 weeks to detect the disease. So the best part, the important thing about diagnostic tests and diagnostic tools is that the disease has to be diagnosed as soon as possible. So the diagnostic test should be such that we should be able to detect the disease, I mean, at the earliest. So if we are able to disease right after a couple of days, the person has been infected, I mean, nothing like that. So we cannot wait for like 2 weeks for the diagnosis of the disease, and by that time, it must have kind of reached, I mean, to a larger population. So we cannot afford that, right? So that's the reason. I mean, the gold standard currently that's with us is the real-time based assays. So when we are talking about real-time based assays, that means we are trying to diagnose the disease with its nucleic acids. So like I said, when we are taking the swab and we expect that in this swab, we have a lot of viral particles. So that means we need to extract the RNA, that's the nucleic acid that this virus harbors. So we need to extract the RNA of this particular virus. And using that RNA, we might be able to detect this virus. So if you have already understood about the nucleic acid, so like I said, there are two kinds of nucleic acids, the DNA and the RNA. So since this is an RNA virus, so we have to find certain tests wherein we can diagnose this, detect this particular nucleic acids. So while we are talking about these nucleic acids, in case you have previously known or you have understood that we have various tests that we can use to detect nucleic acids, and one of the tests is called as polymerase chain reaction, the PCR. So that's something like we are trying to make millions of copies of a particular gene segment of a particular pathogen. So when you're talking about this SARS coronavirus, I would rather in an easier, I mean, terms tell you that we have to detect a particular gene of this particular I mean RNA virus. So for detection of that particular gene of this RNA virus, we have a very simple test that's called as polymerase chain reaction. In polymerase chain reaction, we have certain kind of, I mean, primers, we have certain buffers, we have certain other reagents that we use to kind of amplify and make millions of copies of a particular gene. So we use certain primers that are more specific to certain genes of this virus and try to actually make millions of copies of that gene and diagnose that particular virus. So when we are talking about the polymerase chain reactions, we need to understand the polymerase chain reaction is a kind of conventional test for diagnosing the nucleic acid of a pathogen. And now we have some different tests that are kind of modifications of a polymerase chain reaction, and that we call as real-time polymerase chain reaction. The only difference between the simple polymerase chain reaction and the real-time polymerase chain reaction is that real time means we are doing something, we are seeing something live. For example, we are watching a cricket match live. I would say that I'm watching it real time. So it's happening this time at Lord's Stadium in England. I'm able to watch this cricket match live at the same time, sitting here in Kashmir, right? So watching something going live is something what we call as real time. So the only difference between the PCR and the real-time PCR is that we can see the amplifications, the millions of copies being made real time, rather than seeing the millions of copies have been made after the test has finished. So the only difference between a PCR and the real-time PCR is that we can see things going on live, and in a simple PCR, we cannot see things going on live, right? So so maybe I can kind of, I have a small presentation. I can kind of maybe share my screen and sure. I guess for some reason I'm not able to share the screen over here, so So I think I don't have this. I'm not finding this option of sharing the screen. I don't know what's not here. So yeah, it's a document. So anyways, maybe I'll try to explain it like that. So I'm not here getting the option of sharing the screen actually. So the important thing is that when we are trying to detect the nucleic acids, like I said, we have two tests, the PCR and the real-time PCR, and the gold standard for diagnosing uh, the the SARS coronavirus is the real-time PCR. So in real-time PCR also we have two kinds of tests. One is called as relative quantification test and the other is absolute quantification test. But when we are talking about diagnosing a particular pathogen, we usually use a the absolute quantification test. So absolute quantification test would mean that we'll be able to know the absolute number. So that means the number of viral particles that are present in the patient. Let's say, for example, per ml of the sample collected. So that will also give us an idea what is the virus load inside this patient, what's the kind of load that we can see inside this patient. So, so maybe, so I have a screen over here also. Maybe if you can, if it's possible for you to see, just let me know if you can see the screen. So we have this is a real-time based test, and we can see we have a simple test. What we call as the first test is simple relative quantification test, and the second test is attachment probe test. So usually, what the test that we usually use for this is a attachment probe test that we call as absolute quantification test. So so this is a better, example. If you can see, we extract the viral RNA, and you can see a viral RNA at the top. And once we get the samples, what we try to do is extract the sample. And once we have extracted the samples, what we actually extract it is the nucleic acid of the sample. So the nucleic acid of the sample is actually the RNA. So once we are extracting the RNA, so the important thing is that we cannot do much with the RNA, and the important thing is that we need to convert this RNA into cDNA. So we make use of certain primers, whether they are specific to certain genes or they are random primers that can actually convert the RNA to a cDNA. So if you can see, the viral RNA is slowly getting converted into the cDNA. And once this is converted into a cDNA, we are actually making use of certain primers and also certain probes. So if you can go back and take a look at this particular picture, so we are making use of the primers, the forward primer and the reverse primer, and we are also making use of the probe. So the only difference between the classical real-time or the polymerase test is that the TACMAN probe, the real-time absolute quantification test, makes use of primers as well as a probe. So the probe kind of is a fluorescent probe, and you will see the probe kind of, the primers anneal at their particular places, and the probe anneals right in between the primers. So you can see this is the probe and these are the primers. So once the primers start the amplification, it starts adding the nucleic acids. So the probe contains two kind of molecules. One is the reporter molecule, and due to this molecule, it is a fluorescent molecule. So at the other end of the probe, at the 3' end of the probe, we have a quencher. Quencher would mean as long as reporter and quencher are both present at in the probe, the quencher will keep on quenching the fluorescence of this reporter. So once the reporter is released from the quencher, so you'll be able to see the fluorescence. We'll be able to see the fluorescence would mean we'll be able to see the amplification. So seeing the amplification would mean that actually we are detecting the viral gene is being detected. So here, once the replication starts over here, you'll be able to see the primer keeps on. It's extending, and because it has a 5', 3' exonuclease activity. So the probe, the reporter here will be released. So once it will be released, you'll be seeing the fluorescence. And as we see the fluorescence, we'll be able to understand that something is being, some gene of the virus is being amplified. So this fluorescence we can see real time. That means once the test has started, we are able to see the fluorescence. That would mean that we're actually able to see that the amplification and some amplification of the gene is being, of the virus is being, some gene is being amplified. So that's why it's called as real time. So as of now, this is the kind of gold standard test that we use for diagnosis of the disease, and we don't have any other tests that is as reliable, as specific, and as sensitive as that of real time as of now. So we cannot rely on the antibody test, like I told you, because these tests are kind of, these antibodies are elicited in the host after like a couple of weeks of the infection. So we cannot wait for a couple of weeks. So, but we can always go for serological assays and population screening. For example, if we were to see what's the population that was infected with the SARS coronavirus. So maybe we can go for a random sampling in, for example, Srinagar or rest of the Kashmir, and take blood samples, collect sera, and if we have a proper antibody test available with us, we can go for random screening and see what is the population of this population that was positive for this particular test. So that's a random screening test, and that can be done based on the antibodies. But that's not a test, the diagnostic test, that we need at the time of a pandemic. So we cannot have the test at the time of a pandemic. In a pandemic, when there is an outbreak that's causing huge morbidity and mortality of a host population, we need to understand the test has to be very fast, very quick, sensitive, and reliable, and should be very specific. So like I told you, the real-time assay, so the currently used real-time assay does not take care of just one gene of the virus. So we have multiple genes. For example, we take care of the nucleocapsid protein, the nucleocapsid gene, so that's detected. We take care of the envelope gene, that's detected. We have one gene that detects one part of a non-structural protein that we call as ORF1AB region, 1B region. So at the same time, from a single sample, the patients are tested for this coronavirus at 3 different sites of the virus: the envelope gene, the nucleocapsid gene, the ORF1AB. So if at least there are 2 positives out of these 3 genes, so they might say that this particular sample is positive for this virus. And before they start actually the screening of this virus alone, so they go for a general screening of coronaviruses. So we need to understand that these coronaviruses, this is not the only coronavirus that affects the human beings. So there are a couple of more normal coronavirus, flu-like coronaviruses that cause diseases like some basic flu-like diseases that reside in the nasal tract of the humans. So before going for the specific test, they go for general test whether this patient is affected with the coronavirus. So if there is a presence of coronavirus, they will go for a simple real-time assay, a simple real-time assay, and that will actually detect a simple basic coronavirus. That means either it could be a simple coronavirus or it could be SARS coronavirus too. So that means they will go for a general assay. So then only when the patient tests positive for a simple coronavirus, then they will go for specific virus. Then they will specifically go for diagnosis of the SARS coronavirus too. So that's something that increases the specificity and the reliability of the test. So usually the diagnostic assays, they have to be pretty much, validated. Usually in the other assays, what we do, we go for the real-time based tests, and also these should be validated by the antibody tests. But in this, it's not possible, and we have just one test. That's why the scientists have been trying to increase the specificity of the test. So that's why they have increased the genes that they are trying to target, that they are trying to diagnose, from 1 to 3. So in fact, 4 genes when we are talking about the general coronaviruses. So that's to increase the specificity and reliability of this particular test, because this is the standalone test, the only test that is presently available with us to diagnose the SARS coronavirus. So that's something, I mean, very important. I mean, we do have other tests also available that, I mean, that have been diagnosed. Like I said, these are antibody-based tests that are usually done with a simple, I mean, tests like antibody antigen reaction that we see. So we have a kind of, we use, for example, in those tests, if we are able to, I mean, if we need to go for a population screening and we need to see how many people carry the antibodies against this particular virus. So we need, again, collect the serum samples, because that's where the antibodies are present. So from the serum, we need to go for certain tests, the antibody antigen tests, like simple ELISA. So we do have the serum with us, and we need to have the antigen actually that need to be coated on the plate. So if there is a presence of the antibody in the sera, the antibody will attach to that coated antigen. Let's say, for example, if I talk about the SARS coronavirus, the antibody that we will be interested in, and that gets that's produced in a large amounts in the host, is the nucleic acid antibody. The antibody, again, is the nucleic acid protein. So so that we will take care of that antigen, and we will coat that in a particular, we will have coated plates. And then after that, after some washings, we'll blot antigen, then use our test samples that are the sera, and pour that sera in on these plates, incubate them, and make sure that if there is a presence of that antibodies, those antibodies will get hold of the antigens and bind to those antigens. After that, go for certain washings and have a secondary antibody that will actually tag to the already antibody, nucleic acid antibody that's present in the serum. And with that antibody, the secondary antibody, we will have certain fluorescent substances. We can go for a chemiluminescence or a fluorescent-based assays. And after washing, we can go for a simple OD testing, wherein we can see that particular fluorescence that is present, because that secondary antibody will tag to the nucleic acid antibody, and that nucleic acid antibody that was present in the sera will be attached to the antigen. So it's a simple antibody antigen assay, and wherein we'll be able to detect whether there's a presence of this antibody in the sera, the nucleic acid antibody of the SARS coronavirus 2 in the sera of the patient. So those are the tests, and those tests are a little bit, I mean, they do take time. I mean, when we are developing certain serological tests, so they do take their own time. So they're not as easy as development of a simple real-time based test. So they are protein-based tests, they do take their own time, and with the time, when people keep on working on them, their sensitivity and specificity is increased. So at currently, there are not many tests that are really highly specific and reliable. But there are a lot of tests available that can do a basic screening test. But there is always a possibility of false positive and false negatives when we talk about the serological assays. And the currently available real-time are really, those are better tests for the detection of the SARS coronavirus 2. So I think, I mean, that's all from me. I think there must be some questions, and I need to.
Rostam da, you have board closed? You have board.
So I have got already some questions in this chat. So the first question is, what are the artificial ways to induce the production of antibodies in patients' body we are using recently in Kashmir? What are the antibodies? It's more about it's Farhat Qayoom. So I think it's more about it's more about when we are talking about the therapeutic aspects. For example, so we do have certain. I mean,
now this was a nice presentation by Dr. Nadeem.
Yeah, thank you.
Hello.
Yeah, thank you. I can hear you. For example,
now this was a nice presentation by Dr. Nadeem.
Yeah, thank you.
Hello. Thank you very much, Dr. Saab. This was elaborate presentation on COVID-19 diagnostic approaches.
Thank you so much. Thank you so much.
Thank you very much, Dr. Saab. This was elaborate presentation on COVID-19 diagnostic approaches. Now, I request students that they may pose their questions through text. Dr. Saab will
got a lot of questions. Maybe I can start addressing
students with students every question. I request students they will pose their questions through text.
So can I answer the questions one by one? So I can start answering the questions, right?
Background noise.
So if I'm audible to everyone, I can start answering the questions because I can already see the questions. I can start answering them. So if I'm audible to everyone, I can start answering the questions because I can already see the questions. I can start answering them.
Dr. Nadeem, can you say Zoom chat group? There are
yes,
questions written over there.
I can see and I can start answering the questions. I can see that.
Can you say Zoom chat group?
Yes, I can see and I can start answering the questions. I can see that.
Okay, go ahead.
Okay, the first question is by Farhat Qayoom. What are the artificial ways to induce the production of antibodies in human body we are using recently in Kashmir? So it's more about, it's not a diagnostic approach when we are trying to induce the antibodies. So it's not a kind of diagnostic approach. Maybe maybe we are actually they're actually trying to ask what is the way to induce the antibodies. So we have had certain, certain research going on wherein we are trying to have development of certain, the monoclonal antibody for therapy. So as such, when we have a patient, so it's not like that we can kind of start eliciting then an antibodies against this particular virus. So that's what actually a vaccine does. So if we are able to have a vaccine, and once the patients are vaccinated with a particular antigen, like we have now ChAdOx X from the Oxford, so that has very well been seen to elicit a proper immune response against the particular spike protein, the receptor binding domain of the spike protein. And they have shown that the antibodies are pretty high in number. So those are actually the virus neutralizing antibodies. So when we are talking about the diagnosis things, we're not interested in the neutralizing antibodies that neutralize the virus, but we are interested in any antibody that we can have to diagnose a disease. So like I said, the diagnosis of the best antibody that we can use for the diagnosis of this SARS coronavirus is the nucleic acid, against the nucleic acid protein. So there are no artificial ways to induce production of antibodies. We cannot induce production of antibodies in the patients. But then there have been passive antibody therapy. For example, we use convalescent sera. The convalescent sera would mean sera that from the affected patient. The infected patient will already have antibodies that are neutralize the virus, because the patient who has already recovered from the SARS coronavirus, if he is given those kind of antibodies, that's a proper way to do. And like we have been seeing in other hospitals all over the world, we have been seeing the convalescent plasma therapy happening. So given that the plasma has a good level of virus neutralizing antibodies, that means the antibodies that can actually neutralize the virus, and mostly those are anti-spike antibodies. So the second question that I have is from Sahrish Salim. Is there any chance that the 18-month timeline for development of a vaccine can be shortened? And by how much? So we already had a good news yesterday that the vaccine that has come from the Oxford, that's a ChAdOx X vaccine. So that's not a live attenuated vaccine. So they have pretty much done it very well, and they have used an adenovirus, this replicating platform of the chimpanzee adenovirus, wherein they have cloned, expressed the spike protein of this particular pathogen. And they have seen that after vaccination, it produces a good amount of antibodies, the neutralizing antibodies. We need to understand that there are many antibodies that are elicited against a particular pathogen, against a particular virus, but not every antibody can neutralize, can kill the virus. So there are very small amount of antibodies, and in this case, only antibodies that are raised against the spike protein can neutralize the virus. So that's very important. So when we are talking about the 18-month timeline, and I think it has been already shortened for a very good vaccine that has been developed by Sarah Gilbert of the Jenner Institute at Oxford, and it has already come in production with the company called as AstraZeneca, and they have already had a collaboration with the Serum Institute of India for mass production. So I think this is the first vaccine, and which we really feel it's a very promising one. And they have done some, they have already completed the phase 2 clinical trial, and they are already in phase 3 clinical trial that's being pursued in the Brazil currently. So they have been seeing very good results. Yesterday, paper in Lancet was released, and they showed that this vaccine is pretty much safe and produce a good immune response. So the next question from how fast communicable is this virus, and how can we boost our immune system during this pandemic? So we have been all seeing that how fast this virus is. This virus has immense transmissibility because of its very easy transmission. I mean, the only thing that a person needs to do is inhale, and and inhalation is something very kind of innate. He has to inhale. So only thing that he needs to do is come in contact with someone who's already having the virus. That's why it uses the ACE2 receptor, the angiotensin converting enzyme 2 receptor that's present in a lot of tissues to go into the cells, and that's why the communicable, the transmissibility is very high when we compare it with the MERS or the SARS coronavirus 1. And how we can boost our immune system. I mean, we cannot much do. There have been a lot of people that have been saying that boosting the immune system might do this, might do that. I don't personally think that boosting immune system can really do some wonders. I mean, because there is another aspect to it that is called as immunopathology. So whatever severity happens in this disease is because of the lung immunopathology. So there is no reason of like boosting the immune system. So the only thing that we can do is, I mean, is like stay healthy. I mean, do whatever we have to do. The most important thing is like stop this transmission, break this transmission, make sure that we have masks, make sure that we don't touch hands, I mean, and use sanitizers and use the mask whenever it's possible, and whenever we are moving out in the areas where the chances of virus transmission is more. So I have one more question, is that my question is a bit weird. I request you to answer it. Can compromising on nasal, oral, and throat hygiene help us in any way to protect ourselves from coronavirus? I mean, we cannot talk about hygiene. The only thing we need to understand is that it's not, I mean, the basic hygiene is something we all follow, but it's very important that we keep ourselves healthy, and unless and until we don't, make ourselves vulnerable to the getting transmitted, so there are not much chances that keeping our, the nasal, throat, and other passages healthy won't have much difference. The only thing is that we need to understand is that so the only thing that we need to understand is that we have to break the transmission. We have to make sure that we don't go in places where there is the chance of transmission. We don't go in places where there's a lot of crowd. We make sure that we have that social distancing, and we make sure that we wear the mask every time we feel that we are vulnerable to this transmission. So that's very important. And And yes, I mean, whatever ways it's possible, keeping yourself healthy, not only the oral, I mean, the throat and nasal tissues. So that's something, I mean, we know how to do it, taking a lot of like vitamin C, keeping our nasal mucous membranes intact. And that's something, those are very basic things for making sure that we don't have any viral infection, not only the SARS coronavirus 2. So I hope that answers the question. We have the other question is from Shaista Yusuf. What about allergic patients? Why they are more susceptible to this pandemic? Is it like the virus is already present there, or there are some other reasons? So the allergic patients is, I mean, whenever we talk about the allergies, if you ask anyone, any immunologist, he would see that whoever is having more allergies would mean that his immune system reacts more in a way than when we compare to with others. And because it's a kind of immunopathogenesis, the severity of this coronavirus is more about immunopathogenesis. That means the immune system, the kind of cytokine storm this particular virus elicits in the host, and because of that, actually, we it damages the lung tissues, it damages the other tissues, and having a very good immune system does not. I mean, I don't say that we need to have a basic immune. I mean, whatever immune system, whatever the healthy tips we have been following. I mean, that's enough. I mean, people prone to more allergies, whether they are more susceptible. I mean, I have not seen any kind of research, but there are small, clues. I mean, small kind of indications that maybe the people who are asthma, who already have asthma, who already have like nasal allergies, rhinosinusitis, they need to take a little bit more care. Make sure that their nasal, those mucous membranes are still intact in this pandemic. That's very much true. So moving to other question from Ruxana Maqbool, is there any way to distinguish COVID-19 clinically from other respiratory illness, particularly the influenza? So unfortunately, there are not many differential diagnoses that can completely tell you that yes, this is influenza and this is coronavirus too. So there are not many differential diagnosis things. But then, I mean, you have fever in influenza also, and here also we have fever. We have, sometimes sore throat, dry cough, and the respiratory tracts are involved. So there are not many, really kind of clear line between the symptoms that are caused by influenza and the COVID-19. But we need to, I mean, if there is something like that, it needs to be diagnosed. If you have certain kind of symptoms, these kind of symptoms, it needs to be diagnosed. So unfortunately, there is not a clear differential diagnosis between the influenza and SARS coronavirus 2. So we have another question from Kushbu Jan. Is antibody test more specific? No, antibody test is not more specific than the real time. So as of now, the gold standard is the only absolute quantification or the relative quantification of the real time test, diagnostic test, and that's more reliable. And that's something we can do before the antibody actually starts developing in the patient that's infected with SARS coronavirus 2. Another question is from Zaira Sajad. Should contact lens wearers take special precaution to prevent COVID-19? So there have been certain reports that the virus, apart from, inhalation, it can also go through our through our eyes after it can because those eyes are very soft tissues and it can gain entry inside the eyes. But this is something, we are just 6 months down, in this pandemic where a lot of things are unknown. So a lot of predictions are there, a lot of hypotheses are there, but when there is something like that, it's always good to take care about, when you are talking about the contact lens, make sure that your eyes are really stay healthy and you don't actually cause any damage to your eyes in this pandemic. So wearing glasses is always an addition because there have been always reports about, there have been reports that it can be transmitted through eyes. It can act as a route of transmission. So if apart from protecting your kind of oral and nasal passage, you are protecting your eyes also. So that's always an advantage. So one more question we have from Minan Altaf is that, is corona patient, if a corona patient recovers and tests negative, can he get infected again? So there have been reports in South Korea and some part of the world that corona patients that have been infected with the coronavirus, they get reinfected. But lately there has again been in research when we talk about the reinfections, those reinfections are not very severe. And also it has been reported when the reinfected patients, they cannot really transmit the virus to other patients. So that means there could be a little bit of reaction, reinfection, or maybe there's kind of a reactivation of this virus, the already infected virus. So we don't know as of now. But even if it means reinfection, it's not so severe. Once the patient has recovered, so there could be kind of bouts of maybe some reaction reinfections, like it has been kind of a lot of people have reported from all over the world. But then it hasn't been seen to be really very dangerous. Other question is from Fadila. Insha Shakil, how can we differentiate between a normal flu? I have already answered this question. As we know that COVID-19 has a complex and strong structure, so do you think that one vaccine will be sufficient for whole humanity, or we may need different vaccines from the same virus? So as of now, like I said, the vaccine that has already been developed, the Chadox X from the Oxford, it looks like that this vaccine is pretty good. So why do we need other vaccines, rest of the vaccines? So if I'm able to understand this question correctly, is that one way to answer this question is maybe this vaccine does not elicit a proper immune response, and maybe we need a booster dose. So that's something we need to really see when this vaccine comes in a population. We vaccinate a lot of population, and whether we are really able to have that kind of immune response that it stops the infection completely. The other way to answer this question is that maybe this is a virus, it is an RNA virus, and we can have a lot of strains. As of now, there is no strain of it's in just only one strain, because there is no other strain. So a lot of people that are saying that there is a different strain in Kashmir, there is a different strain in Bazaar. There's nothing like that. A strain is something that has different biological types from the other kind of strain. So as of now, the biological features and types are similar. We don't have any strong differences between them. So as of now, there are no strains. But once this this virus evolves, there could be a lot of strains, different strains. So maybe this particular vaccine, again, is this particular strain might not protect us from the other strains. So very well in the near future, we might need certain vaccines that are better modified, that have cross protectivity, that can cross protect us from other strains also. The next question is, how is plasma therapy effective against COVID-19? I have already told you that when plasma therapy, that's some kind of convalescent sera that we get from the infected patients. In the infected patients, when a patient recovers, why does it recover? Is because that the antibodies, the neutralizing antibodies that can neutralize the virus, that can kill the virus, that are usually again is the spike protein of this virus. So they develop a pretty high titer of this virus. Maybe because of the immune system they have, and because they already have this antibodies with them. So that means they once the patient has recovered, they already have this antibody. And if we are using this plasma in an infected patient with a similar kind of blood group, so blood group compatibility is very important in this. That means their blood groups should be compatible. So because they have already have the neutralizing antibodies present in their plasma, and when that plasma is injected in an infected patient, so it can take care of the viral replication. It can kind of restrict the virus from infected more number of cells and other tissues. So that's how it can be effective against the COVID-19. So yes, one more question is that, is hydroxychloroquine sulfate tablets useful for COVID-19? And there have been a lot of reports right from the start of this pandemic in February only, when they found that the combination of remdesivir with chloroquine was found to be effective. But why chloroquine and other drugs have been found is, we call it something like drug repurposing. That means whatever has been regulated, whatever the FDA and other regulatory organizations, drug regulatory organizations have already passed for other diseases. Let's say, for example, a paracetamol and other drugs that we already find in the market. So the regulatory bodies, the FDA and other regulatory bodies, have already confirmed them that they can be used for they can be used for in the human population against certain diseases. So that's what we call as, if we are trying to see whether at random whether any drug can control coronavirus, so that's called as drug repurposing. So because this is a pandemic and we do not have an option, we do not have a vaccine, that's why people all around the world were trying the already available drugs that have been cleared by FDA and trying to use them against coronavirus. And using hydroxychloroquine was just a random try from certain people, certain researchers, to see whether it has an effect. And unfortunately, in some research, they found that it does have, I mean, a good effect. But in other, subsequent studies, it was found that it does not have a very good effect. In fact, hydroxychloroquine is not at all safe to take. So it was promoted by certain organizations, but then there were a lot of retractions of certain papers that were saying that hydroxychloroquine is really effective, and that was actually, I mean, those there were a lot of doubts. And then I don't think nowadays anyone is talking about hydroxychloroquine. It's something, I mean, people have like kind of already forgotten. So a new paper was about, in fact, about dexamethasone. So in UK, there was a big study that they say that it does have a good effect. So as of now, the only antiviral that clearly affects the virus, that stops the virus from replicating, is remdesivir. So that's currently the only antiviral that's been seen. And there are some other antivirals also, like favipiravir, that was kind of in a research in Japan. People found that that's a very effective drug against this. But as of now, we don't have a really promising drug, including remdesivir, that can stop this virus. So because it's a very complex disease, it involves your immune system, it involves a virus replication. So a lot of things are going on when this really starts causing the symptoms and the severity of the disease. So as of now, we don't have a good drug that we can say, okay, this is the disease and this is the drug we can really give and protect this virus. So these are all kind of more like symptomatic, or maybe they can reduce the severity of the disease. So that's why these drugs are given. So for moderate infections or less severe infections, so they only recommend you simple paracetamol and other things, and they advise you to be isolate yourself at home and don't infect others. So that is the only thing they advise. So we have one more question is, vaccine will be effective in 3 months. Okay, so I have read that vaccine will be effective only for 3 months. Or can you please, there have been a lot of studies that they say the virus neutralizing antibodies that are elicited by this virus, they don't stay for much. They kind of fade away in a couple of months, 1.5 months after that. You see that these antibodies start fading away. There are studies about that. So as of now, we really don't know whether this vaccine will produce the long-lasting immunity, or maybe we will need a continuous booster doses of these vaccines to make keep the production, keep that particular. As of now, we really don't know whether this vaccine will produce we have long-lasting immunity, or maybe we will need a continuous booster doses of these vaccines to make keep the production, keep that. As of now, we really don't know whether this vaccine will produce a long-lasting immunity, or maybe we will need a continuous booster doses of this. So I'm finding that echo from somewhere. Anyways, what we have one more question is that if the vaccine developed proves effective, then how long will it take to eliminate the virus from the whole world? So we cannot talk about the whole world, but it's like how effective the vaccinations are, how effective the vaccination procedures are, and whether the vaccines really reach everywhere. So if vaccines have a long reach, I mean, they reach really every person, who is susceptible. So we have to remember that we have to vaccinate the people who are vulnerable, the healthcare workers, the people sitting there in the grocery shops and or other place, whoever who has a lot of public dealing. So they are the first vulnerable person. So the vaccination has to go in phases. The first phase, second phase, and third phase, and hopefully it can reach the maximum population. So the more reach is there, so there's more, possibility to end of an end to this pandemic. So people suffering from gastric or duodenal ulcerations, do they have more chances of coronavirus? So there is, this virus has a problem of what we call as tissue tropism. So it does not only infect the lungs, we have seen that it infects the nervous system, the gastrointestinal infections. The symptoms are very well kind of manifested in this particular virus. We have been seeing the people, one of the importance is the manifestations, the symptoms is diarrhea, vomiting, and if your gastrointestinal tract is really, infected, you feel that you are having a long, those infections are really kind of consistent, the gastrointestinal infection, the diarrhea and vomiting. So it can very well mean in this pandemic that it could be SARS coronavirus. And at this point in time, it's really important to keep all our mucous tissues, membranes very intact. Ulcerations, maybe it can lead to kind of make you more prone. So whatever kind of makes you prone to these infections. But the important thing is how well you are able to control yourself, wear mask, and make use of all the standard operating procedures, all the SOPs. So that's more important. The susceptibilities come. That's something that comes next. When you are exposing yourself, then only you feel that you are at What about the claims of Oxford University vaccine development? How much time will it take to be available to common masses? Like I said, it will not take much time. The vaccine development has already kind of started. That's it. We have already had the vaccine that's already, in mass production now in phase 3 clinical trials. I think it should not take much time to get the vaccine in the market. So how much protection the mask offer? Mask as of now offers you a complete, not complete protection, but the best protection that is available as of now with us. So yes.
Okay, Dr. Nadeem, this was a nice presentation. I think students.
Okay, yeah, I got it.
Okay, Dr. Nadeem, this was a nice presentation. I think students. Okay, thank you very much.
Yeah, thank you so much.
Many are background. Can I lie?
Okay, thank you very much.